Hello, everyone. My name is Melanie. I am a senior at Johns Hopkins, double majoring in chemistry and biophysics and minoring in environmental studies. And for this presentation, I will be talking about serine threonine tyrosine kinase 1, otherwise known as STYK1, and why this protein sticks out as a receptor tyrosine kinase. So first, I'm going to go over the general mechanism of activation for receptor tyrosine kinase proteins. And what you see here is a depiction of the plasma membrane. And there are two monomer RTKs identified as trapezoids, and there are two ligands represented by triangles. So then you have the ligands bind to each of the monomers, and they come together forming a dimer. And this dimer is then capable of cross-phosphorylation and adding a phosphate group on each of the tyrosines on the monomer. And lastly, this activated dimer is able to bind to other proteins that are involved in the resulting signal transduction pathway. And here are all the different RTK families, and you can see they all follow the same structural motifs of an extracellular domain, a transmembrane domain, and an intracellular domain. So here is STYK, and you can see STYK is unique because it lacks this extracellular domain, and therefore it does not require ligand binding to induce activation, which is very rare for RTKs. And this protein is also known to be constitutively active. And previous researchers have hypothesized that this is because of STYK1's high propensity to aggregate. STYK1 is also found to be highly overexpressed in cancerous tissues, seen here on the right. And another interesting factor is that it is present in castration-resistant prostate cancer. So even after complete surgical removal of the prostate, this cancer is still present, thus proving that rather than merely surgical interventions, therapeutics targeting RTKs such as STYK is also needed to battle these diseases. So the first thing we sought out to discover is what is the oligomer size of STYK itself? And to do that, we collaborated with a different lab, Stoneman et al., and came up with an analysis method called Fluorescence Intensity Fluctuations, otherwise known as FIF. And how this works is we transfect a CHO cell with 2 micrograms of STYK DNA linked to a fluorescent protein YFP. So we take images of these cells on a confocal microscope. And then when the images are done, we begin to analyze them with the FIF software. And the first step is to create a region of interest, an ROI, around the perimeter of the cell, which you could see here in red. And next, this program segments this ROI, this region of interest, into 15 by 15 pixel segments. And from each of these segments, there is a brightness histogram created. From then, a variance and average is computed. And the result is all of these segments and their individual histograms are combined to form a resulting one-dimensional brightness spectrogram. And you can see here on the right our STYK1 data. And what this reveals, this is fitted to a Gaussian curve, and the probability of one lands around a trimer size. So this supports the idea that STYK1 itself is a trimer. However, we did run into a problem because our cell images are very heterogeneous in the way that some of the segments had really bright spots and really dimly lit spots. And what happens with the first algorithm was that when we had a really bright spot in the segment, all of the dimly lit spot data would be discarded, and therefore it would be skewed and not analyzed. So we collaborated and worked on a despotting algorithm. So this results in images of cells in which one contains the dimly lit spots in the segment and one contains the bright spots in each segment. And therefore the normal FIF process continues normally for each of these data sets independently, and then we are able to compare them. And here is an example. So this is a cell, and our ROI here consisted of the cell minus the nuclear region. And after going through the despotting process with a threshold of 1/2, we see bright spots removed and analyzed separately. So when we look at the data, we notice both the spots, the really bright spots, and the not so bright spots, when both analyzed independently, reveal trimers as of the oligomer size. So this further supports the idea that fifth is a trimer in its oligomer state. We then set out to look at FSI FRET data, and what we saw was barely any FRET interactions, and our fluorophores were placed on the C terminus of the kinase domains on each monomer. So this shows that where these fluorophores are, they have to be at a distance greater than 100 angstroms to account for this lack of FRET interaction. And this is also rather unique, since we usually do see FRET for RTKs, as you would expect the monomers to be close together in its oligomer form. So we put all of this data together to create the first model of STYK1 in its trimer oligomer state, which you could see here. There are 3 monomers shown in pink, yellow, and green. And when we look at it from a different angle, we could see here at the ends of the kinase domain C terminals, they are all greater than 100 angstroms apart. So this fits in with this constraint, along with the fact here shown in black are the catalytic domains, and they're all close to each other to allow for cross phosphorylation to occur. So all of our restraints are accounted for in this model, which leads me to our conclusions, which are that we presented the first model for STYK1 in its oligomer state, and it meets the requirements that our fifth data revealed for STYK1 being a trimer in the plasma membrane, the lack of FRET data suggesting that those areas are far apart from each other, and that the catalytic domains are close enough for phosphorylation activity to occur. Another important finding is that because the despotting data revealed to be a trimer for both the bright spots and the dimly lit spots, this disregards our initial hypothesis that the bright spots are higher order oligomers, but rather these are just STYK1 rich domains in the membrane comprised of trimer aggregates. And lastly, all of these lead us to understand better STYK1's implication in cancerous tissues, because it is constitutively active and it doesn't have its ligand binding domain. So it is growth factor, and it exhibits growth factor independent activation, which leads to perpetuate cell proliferation and mitosis, leading to tumorous tissue. That is the end of my presentation. Thank you so much for listening.